of the cell membrane. In this episode, we're going to be looking at the movement of materials through the plasma membrane. So transport of materials is super important to cells. They have to get materials in so that they can use them. They have to get rid of materials. Sometimes that might mean waste products. Other times it might be some product that they make. So the membrane is going to allow for all of that, but it's selective. It doesn't let anything easily pass through, and therefore we call it a selectively permeable membrane. This is going to allow certain types of fluids to move easily. And we say fluids here, very often what we're really referring to are different things in solution, right? So solutes dissolved in solution. One example of that is that cells actually try to maintain an imbalance of sodium and potassium ions on different sides of the membrane, and we'll see why that's really important. Other things we will consider here is that the transport can happen in one of two ways, either passively, which requires no energy, or actively, which means it's going to require the use of the energy molecule ATP, which is the most important energy molecule used by cells. Passive transport is movement through a membrane passively, therefore no energy is required, and the way in which particles pass through the membrane is a process called diffusion. So diffusion occurs when there's a particle that can pass through the membrane but is in high concentration on one side of the membrane, and if it's small and if it's nonpolar in particular, it's going to be able to pass through the membrane just on its own. So here, kind of think of it as here's a lot of particles on this sort of time scale here. So here's the beginning. You see lots of particles that are in fluids out on the outside of the cell. So that's extracellular fluid. Now a little bit later, because there's so many of these, some of them are able to pass through. And so they start to pass through here. You see there's some now inside the cell in the cytoplasm. And then ultimately passive transport will end up with this state we call equilibrium, where you're going to have approximately the same amount of particles on each side of the membrane. It doesn't mean that none are going in or none are leaving. It just means that there's no net movement. So you have kind of this relative balance here. The molecules that can move best through in this way are the really small ones and the nonpolar molecules that can pass through. So oxygen and carbon dioxide and lipid hormones can diffuse easily through the membrane going either way. So that's something to look at. Other factors that are going to affect how the rate of diffusion, how something can move through, it's going to have to do with how concentrated it is on one side, or its concentration gradient. The more difference there is on one side than on the other side of the membrane is going to lend itself to faster diffusion. The mass of the molecules, okay, the smaller they are, the more easily they can move. Temperature, the warmer the temperature, you're going to get faster movement of particles. You remember from chemistry that the particles will have more kinetic energy. This solvent density, so how, and you might think of this as almost thickness, but it really has to do with how dense there is, the number of particles that are dissolved in, for example, the cytoplasm. And then we have solubility, how more there are more nonpolar lipid soluble molecules that can pass through. Distance traveled, surface area, all of these things have an influence. And then the last one, pressure. So in some environments in the body, for example, like in the kidneys, there's a difference in pressure that will make a difference in how quickly something can pass through membranes. Facilitated diffusion is the passing, still passively, not using energy, of materials that are going to go through the membrane, but they are going to pass through something that helps them. In this case, it'd be some kind of a channel that would help them to go through. And so the proteins we looked at earlier, the ones that can actually span the entire membrane, these transmembrane, integral membrane proteins, some of them are channel proteins that actually, it's kind of like a little slide that's going to allow something to pass through without having to contact that lipid bilayer at all. And there's two types of facilitated transport proteins. One are these ones I just mentioned, which are called channel proteins, kind of like the slide. And then there are some that are actually designed to facilitate movement across the membrane. They're called carrier proteins, but it's still not using energy. So we'll take a look at that in just a sec. So channel protein, here it is. You can see here the little slide affair that we have here that's a protein that serves as a channel for certain things to pass through. Very often these channels will serve to allow certain ions, which have a charge, remember, or polar molecules, to pass readily either in or out of the cell. Some have gates on them, and some are open all the time. An example of these would be aquaporins. So the word aqua you see is water, and porins like pore. And these are channel proteins that are very specific to allowing water to come in and out, to move in and out. Muscle cells also have these channel proteins, but theirs have gates on them, which is going to be in response. These little channels will open up in response to some activity, some signal. Carrier proteins, actually, they change in shape and allow things to pass through. Still no energy used, but something on one side of the membrane, here's the extracellular side, we have some fluids, but we have some solutes here. In this case, we're looking at glucose. It can bind on to the outside of one of these carrier proteins, and once it does, then the carrier protein actually changes shape and allows it to kind of move through, and then it changes shape on the other side, allowing that glucose to come into the cytoplasm, and then it opens up again for the next one that can come through. So this will help allow certain things to come through more readily that wouldn't normally come through quite that easily. And these can work in both ways. They can take things from the outside and move them into the cell, or in some cases they can take things from the inside of the cell and move them out. Now with glucose, the cell wants glucose, so it's generally moving things into the cell. And we'll talk about glucose in a little while. When we use the term diffusion, but we're talking specifically about water, there's a term that's used called osmosis. So osmosis is diffusion, but it's diffusion specifically of water across a membrane. And just like the particles, water is going to move from where it has a higher concentration of water to an area where there's a lower water concentration. And these differences in concentration are going to make a difference, or the amount of solutes that are dissolved on one side or the other are actually going to affect the flow of water from one side to the other. We're going to do some practice with this coming up. As a matter of fact, one of your gizmos is going to look specifically at osmosis. So stay tuned on that. The term tonicity, so tone, we all know what being toned is. Tonicity is really describing how that solution, how some solution from the outside, so that's extracellular solution, can have an effect on how the cell volume is. In other words, how filled up that cell is, how much water is able to come into that cell. So there's tonicity is related to something called osmolarity. And osmolarity is referring to, so it's kind of like this refers to this, and this is referring now to the total solute concentration of a solution. So it's looking at all the things that might be dissolved in a solution, and some of them are going to be particles that might be permeable, and some are being allowed to move through through, and some are non-permeable solutes, which means that they're stuck and they won't move through, probably based on size or charge or both. So when solutions have different osmolarities and they get separated by the plasma membrane, then what you're going to see is that the water is going to move, not the solutes. And so we'll see this in a couple of demonstrations, but water is going to move from whichever solution has the lower osmolarity, in other words, it has less dissolved material in it, to the other side where there is more dissolved stuff but less water. So these terms, hypertonic, isotonic, and hypotonic, are descriptors of the osmolarity of the cell compared to the surroundings or the extracellular fluid. And these are terms you need to know, so we want to pay attention to these. Here's an example, a classic picture of it. So these are red blood cells in all of these little images below. And when we look at a solution that we say is hypertonic, meaning we're looking at what's outside the cell. So if this group of cells here is in a solution that is hypertonic, meaning it has lots of dissolved materials in it, then water's going to move out of the cell. It's going to move this direction, out of the cell. And what you see here, the result of that is, look how these cells, these red blood cells that were normally kind of disc-like, have now started to shrivel because water is leaving them and going out. Here, in an isotonic solution, these red blood cells are sitting here, and something that is iso, meaning the same, has the same dissolved solutes outside as inside the cells, then there's kind of an even exchange of it, and the cells stay their normal size and shape. But if you put the group of cells into something that has really a hypotonic solution, meaning there's almost no solutes in this out here, might think of this as sort of like distilled water, something that would have almost nothing else in it but water, then what happens is the water moves into the cells. It moves from that area where the extracellular fluid has this really high osmolarity, and so it's going to move into the cytosol. And what happens is you get these really fat-looking cells which actually can explode. So when we're looking at animal cells in particular, the best thing for an animal cell is to be in an isotonic solution. So kind of think saline solution. When you put it in your eyes, for example, if you've ever used that, it would be isotonic to the cells in your body. So, but plant cells have a different thing in that they have these walls, and cell walls are useful. They also have, remember, that large central vacuole. So if a plant cell is in an isotonic solution, it's going to just have this pretty even exchange of water in and out of the cell. In a hypertonic solution, where the material on maybe out here would be a lot of salt or other things dissolved, then the water is going to flow out of the cell, and that cell is going to shrink up and plasmolize. It's going to get really shrunken. In a hypotonic solution, meaning we're putting this cell in a water that is a lot of just water, look what happens.